I'm very pleased to introduce Professor Dalton Conley. Dalton is a Henry Putnam University professor in sociology at Princeton, where he is also affiliated with several research centers. His research spans a wide array of topics, all evolving around the question of how socioeconomic status and health are transmitted across generations. Academic scholars understand the importance of interdisciplinary research, but most of us can barely master one discipline at a time. Professor Conley's work is truly interdisciplinary. He holds 2 PhDs, one in sociology from Columbia and another in biology with special focus on genomics from NYU. In recent years, he is working on the interplay between health and biology on the one hand and social position on the other. In his current work, he is studying how genes and environments interact. To this end, he uses survey data that includes both sociological, economic, educational, income, other measures, as well as DNA material on respondents. DNA material. In Israel, some would find this unbelievable. Professor Conley is a very prolific scholar, and his papers appear regularly in the most selective and visible journals, including Science, Nature, Context, and many, many others. His main books have been published by top publishers, including UC Press, Blackwell, Princeton, and more. Professor Conley is a research associate at the prestigious NBER, a member of the prestigious Sociological Research Association, a recipient of fellowships by Guggenheim, Robert Wood Johnson, Russell Sage, and more. However, Dalton combines innovative and interdisciplinary basic research with a strong interest in policies that can affect inequality, opportunity, and mobility. He is also committed to the dissemination of scholarship to the lay public. I urge you to check out his TED lectures, web lessons, and the interviews that he conducts with interesting scholars. I use these, my students can attest, I use these materials regularly in my own teaching. So with no further ado, Dalton, please.
My talk will start with how I got into this issue of the interplay of genetics and environment in determining educational success. Then I'll talk about some of the specific research I'm doing now, what's called the Polygenic Score, and then I'll give some examples of how we can apply this new tool called the Polygenic Score to important questions about society. And that is the changing landscape of inequality in the United States, and then specifically how genes and environment interact, and then finally how you can use genes to study purely environmental effects, which I think are another important possibility that is opening up with this new research. And finally, I'll talk about some policy implications, and again, disagree with Avi's opening remarks that just because something's genetic implies that there is no policy that you can apply to that particular outcome. So hopefully we'll have, and hopefully that'll come up in the final panel as well. So the mid-1990s was a very strange time in the United States. And I apologize also that all my data come from the US. When I was finishing graduate school, the right wing took over the Congress. The president signed the end of welfare in the United States in 1996. And in 1997, this book came out, challenging my whole worldview that I had learned in graduate school in sociology. And this is by a sociologist who's Professor Heckman's colleague at the Harris School at the University of Chicago. And she did a number of tricks, I would call them tricks, but very statistical innovations to show that what sociologists like I had been doing before grossly overstated the impact of family income on children's life chances. For example, she would look at the family income when a kid was ages 17 to 21 and saw that income predicted their age 16 test scores almost as well, if not as well, as their income prior to age 16. So unless you have a time machine, the age 17 to 21 is not causing the age 16 test scores. She also showed that all income is not created equally. Namely, if you get an additional dollar from welfare, if you're at the low end, or if you get an additional dollar from inheritance or investments at the upper end, that has less of an impact on children than a dollar from labor market earnings. I could go on and on about this book, but the point was that she was suggesting that there was something besides the income itself, that the income was acting as a proxy for, that was more important in determining children's life outcomes. Now, that could be one of two things. That could be cultural transmission through the family, that certain families that have success in the labor market have a set of values and cultural practices that also breed success in their offspring, and in the household they transmit that. It could also, however, be that there's genetic basis to ability that leads to parental success in the labor market, hence high income, but also leads to offspring success in school and in life. And that is creating what we call a spurious effect of family income on children's outcomes. Now, I should say, for intellectual update, that there's been many studies since in the 20 years since this book was published that have, in a sense, rehabilitated the effect of parental income. Lottery studies in Scandinavia, studies of windfalls of Native Americans, other natural experiments, and including some actual experiments that are going on right now in the United States that suggest that income does have an important causal effect. But this book certainly changed my view that I could not just contrast, for example, poor families and rich families with maybe a couple other factors taken out of the equation, and then say, though, this is the true impact from a policy perspective of income. So for the next 10 to 15 years, I kind of avoided the topic of the effect of family income on children's life chances and looked at other social factors where I knew there was a natural experiment, where I knew that I could really look at a treatment and control group and say that this is really causing the outcome for children. But then in the early 2000s, or really with Clinton's announcement in 2000 of the decoding of the human genome, we had a chance to finally open up the black box of DNA and see how much does this second hypothesis, that is the genetic ability of parents that is transmitted to offspring, that create the spurious effect across generations of income. And that was a result of the genomics revolution and the fact that genotyping, getting DNA information about respondents, became incredibly cheap. It's much more cheap today to take everybody in this room's saliva and tell you about your DNA than it is for me to do an extensive social survey of each of you. So the result is that a number of national studies in the United States and in other countries have now added, rather than starting from scratch, but these national studies have added collection of biospecimens, namely saliva, to analyze people's DNA to better understand the interplay between genes and the environment. Also, in the mid-90s, in our strange time in the United States, the Bell Curve came out, which I'm sure many of you have heard about because it was a bestseller in the United States, a runaway bestseller. And in this book, the authors, Herrnstein and Murray, made a very, I would say, slick, we would say, or sly, but pernicious argument that actually across the 20th century, the United States and other developed countries, but they focused on the US, had achieved meritocracy, had actually taken away all the inequalities that we're talking about today, and that the resulting distribution of incomes or of educations was purely the result of merit, purely the result of ability, because the old barriers, quota systems in higher education, the old boys network as they call it, and so forth, had fallen away in this magical land of mid-20th century America. But the perverse irony, they argued, was that because these inequalities, these remaining inequalities were based on innate ability, they were more intractable. They were more resistant to policy interventions, as Avi was saying, and they were also fair because they were based on ability, and therefore we should not be concerned with social policy to rectify inequality. Now, I'm not going to spend a lot of time beating up on their argumentation because many others have done it. I'm going to talk about their data a little bit for now. They used what was called the National Longitudinal Study of Youth, 79, and that included a birth cohort of students they followed from 1957 to 1964. That's 7 years. They're making a sweeping argument about social change across the entire 20th century based on a group where their birth cohorts only span 7 years. And in that 7-year period, they showed that the economic return to IQ, which is measured in the NLSY or a proxy for IQ, had gone up. So from these two facts, that the effective IQ over these 7 years had risen, they concluded this meritocracy is here and all inequality is the result of genes. Now, of course, there's several problems. I already mentioned that the time span is too short that they analyzed to make those kind of claims, but also they measured cognitive ability in the survey, but cognitive ability, we know, has both an environmental and a genetic component, and therefore it's very possible that over those 7 years, it's the environmental component of IQ that's become more important, or its association with family background and differential parental social class. So we went ahead, now that we actually have genetic data, and we measured the genetic predispositions for education of respondents, and then we looked at a much wider group of the right birth cohorts born from 1920 to 1960, and we asked some of the same questions that Herrnstein and Murray asked in the bell curve, and I'll tell you what we found. Let me grab some water, sorry. So this is a very short, I promise, 1-minute biology or genetics lesson. We all have, if you take your 23 pairs of chromosomes and unwind them and lay them end to end, they're about 6 feet, just shy of 2 meters tall. I think that's a coincidence that they're about as tall as humans are. It's not a divine intervention. And there's about 3 billion individual base pairs in your DNA. When I say base pair, I mean one of the 4 bases, T, C, G, or A, and its complement on the other strand of DNA. Of those 3 billion, about 3 million very commonly in humans. Hence, you hear the term that we're all 99.9% equal, because there's 3 million, that's 1 in 1,000 locations where you see variation. So what we focus on is the most common variation among that, which is about 1 million. You can then, I won't go into it, but you can then infer the other 2 million and get up to 3 million locations where you and I may differ, or even me and my full sibling will differ, 50%. So what do? We take each of those locations that are measured in the data and we add them up. So at the first location, we call this the A allele. So let's say 10% of the population has A there, or 10% of the alleles are A, and the other 90% are T. If I have the minor allele, if I have one, because we have 2 chromosomes for each allele, I might have 0, 1, or 2 A's there. So I hope everyone's following me. At the next location, we call T the one that we're going to score. And we do this 1 million times or 3 million times, and we run 3 million little statistical tests. and we want to know the effect of each of those alleles on your educational attainment, how many years of schooling you completed. Then we do that in a very large sample. So what I'm going to show you includes analysis of almost 1.2 million people, because you need a huge data set to detect these very small effects. Then whether or not they seem to be big or small, we take those results, what we'll call the betas, the weights, the result from that experiment, that statistical test, and we take it to a new sample. So we're not having just confirmation in our existing sample. We take it to a prediction sample, we call it, a replication sample, and we then score everybody in that sample. So if I have 0 A's, then I get 0 times negative 0.082 for that location. If I have 2 T's at the next location, I get 2 times 0.064 at that location. Do that 1 million or 3 million times, and I add it up, and the result is a polygenic score, a single number that represents your genetic potential for educational attainment. That sounds really scary, that there is a single number, and I'll come back to that at the end and the policy implications for that. But for now, I'm going to use that as a tool to talk about shifting environmental influences. So just to talk about, these are the 2 studies I'm going to use. Like I said, there was almost 1.2 million people in the training of the data, but this is where I'm going to apply those results. One is a group of adolescents that were followed into adulthood, called the National Longitudinal Study of Adolescent to Adult Health, and the other is a complementary study of older Americans called the Health and Retirement Study. So I'm going to skip all this stuff and show you what it predicts. So if you look at the far left, you see that in the National Study of Adolescent Health, the best predicting equation, the best predicting number, predicts 13% of the variation in the population of their educational attainment. And even though we never measured cognitive performance in the 1.1 million people, because education and cognitive performance are so related, we can predict cognitive performance with an 8% R-squared. That is, explain 8% of the variation in the entire population. And it's a little bit lower for the Health and Retirement Study on education. It's a little bit higher on cognitive performance in another study called the Wisconsin Longitudinal Survey. But just to put this in context, so this is what this score predicts. If you're the bottom quintile, then you have about a 10% chance of graduating with a 4-year college degree. If you're in the top quintile, you have a 60% chance of graduating with a 4-year college degree in the United States. I can predict a 5-fold difference based on your saliva. Or now if you allowed your imagination to go forth, or based on a baby's saliva, or based on an embryo's cells. I'll just let that sink in as we go forward. To put it in another context, to give you comparative perspective, in the dark green is that polygenic score I'm talking about, predicting about 10% in this rendition, and it has almost as big an effect as your mom's education, which in terms of socially measured variables is the single biggest effect. If you combine both parents' education, you can see on the far left that you get a bigger effect, but it's almost twice as big in the final rendition as the effect of parental income. So this is really important, I think, that if you're going to do analysis of social policy or of educational policy, if you're going to do an early childhood study, if you're going to even reanalyze the old early childhood studies like preschool, if you take out this effect, you're going to see the effect of your intervention much more starkly. You're going to see it less biased and more precisely estimated. So even if you look within families, So the far left bar is the original score, and the far right bar in all three groups is how it predicts between siblings who share their common environment, share the parental influence, but differ because they're only 50% related on their polygenic score. So you see that even within families, between siblings, this predicts. So it's not just picking up some sort of ancestry effect or some cultural effect. It's truly the random assignment of your alleles, of your genes at conception that has its effect. So I'm going to address three, using this score, actually an earlier version of this score, I'm going to address three central claims that Herrnstein and Murray made in the bell curve. And the first is what I mentioned, that social stratification is increasingly giving way to genotypic stratification. And I quote them, "High cognitive ability as of the 1990s means more than ever before that the chances of success in life are good and getting better all the time. And these are decreasingly affected by the social environment, which by extension indicates that they must be increasingly affected by genes." So let's test that. So if you look at body mass index, how much you weigh for your height, you can see, so the top line is somebody who is one standard deviation above the mean in this kind of polygenic score I was just talking about, but one that's trained for predicting overweight or obesity, for predicting body mass index. And the bottom line is someone who's one standard deviation below the mean. So you see that if you're born in 1920, someone who is, has a higher polygenic score is indeed more likely to be heavier, but the gap is small, right? And then if you're born in 1960, everybody is heavier. As you can see, both lines are going up, but the gap is also bigger. There's a significant gene by environment interaction over the course of the 20th century, changing the determinants of body mass index, such that genetics has become more important in determining who is overweight and who is thin. That would make sense, right? Born in 1920, you're growing up in the Depression. There's not a lot of calories around. There's probably, everyone does more physical activity, but the moment you enter the late 20th century, calories abound. Anyone can eat anything they want. Food is cheap and abundant. The nature of the economy has changed, so not a lot of people work outdoors physically for their livelihood. So you start to see, well, 1,000 genetic flowers bloom. Everyone reaches some sort of different level of body mass index. We see a similar effect for smoking, which I won't show, that after the negative impact, the true health impact of smoking became known, genetics became much more important, because only the really hardcore addicts of nicotine, essentially, their nicotine receptors in their brain are hardwired to really need nicotine once they experience it, are the only remaining smokers. The genetic effect has gotten more and more salient as the number of smokers has decreased. So you might expect that Herrnstein and Murray then are right about education. As we expand educational opportunity, as education becomes more common, higher education, that we see the genetic effect get bigger. That is their argument. But instead, we see the opposite, actually, in the United States. We see that over the course of the 20th century, there's a very slight narrowing of genetic gaps in educational, how genetic differences translate to genetic gaps in educational attainment. So strike one for the bell curve. One of their second central claims is that spouses are sorting genetically to a greater extent more recently. I'll read their quote again. "Add to this phenomenon known as assortative mating. Likes attract likes when it comes to marriage, and intelligence is one of the most important of those likes. When this propensity to mate by IQ is combined with increasingly efficient educational and occupational stratification, assortative mating by IQ has more powerful effects on the next generation than it had on the previous one. This process, too, seems to be getting stronger, part of the brew creating an American class system." So if you have people sorting on their genetic ability for education, then in the next generation, the standard deviation or the variation, the gaps are going to be wider just because of the that's also a scary thought. And indeed, in early work we saw that spouses are similar. This is in American data. If you just look across the entire genome, we like to think in a modern society that cousin marriage there's a strong norm against cousin marriage, but by accident, people are marrying people that are equivalent to their first cousins once removed on average. And if you factor out their ancestry, their sort of ethnic group, that it's still second cousins. So lest we feel superior to other cultures where there's actual first cousin marriage, we're not doing so much better. But that doesn't tell you the correlation on the genes that matter for education. That's just across the entire genome. So we looked at that. We looked at the correlation in polygenic score for a number of outcomes, and you see that on the left bars, the light bars, is the spousal the correlation between spouses in the actual measure, in what we call the phenotype. So actual years of schooling for spouses is correlated above 0.5. Height is correlated at about 0.18. Body mass index about 0.24. And depression at about 0.3. But when you look at the genotype, what's the correlation on the underlying genetic signature of that outcome, you see that height acts very weirdly. It's actually stronger for height, but for education, it's significant. It's not zero, but it's much lower. It's in the order of 0.14. And for BMI and depression, it's nothing. But their claim, so there is some sorting based on educational genotype. However, their claim is that it's been getting more so over the 20th century. If you look at the phenotype, it is indeed significantly increasing. But if you look at the genotype, there is no trend whatsoever in the level of sorting by genotype for any of the four depressions.
That you explain the difference between phenotype and genotype.
The question is, or the comment, the suggestion is to explain the difference between genotype and phenotype. So phenotype simply means trait. I don't know how this is being translated into Hebrew as I'm speaking, but it means trait, it means outcome, it means how much you weigh, how tall you are, how many years of schooling you have, how you score on an IQ test. Genotype is the underlying genetic signature. It's not a single gene. It's what I'm talking about in this case, that number that captures the effect of 1 million different locations in your genome. That's the genotype. I hope everybody got that. I would normally say there's a sociology norm, which is you don't say anything until the end of the talk, then you ask your questions. And there's the economics norm, where you're allowed to interrupt from the first slide. But since this is Israel, I'm not going to allow, I will go with the sociology norm because I don't think I'll ever finish the rest of the talk. But Yossi is accepted from that rule. So third proposition is that we are seeing reproductive stratification. The professional consensus is that the United States has experienced dysgenic pressures throughout either most of the century or all of the century. Women of all races and ethnic groups follow this pattern in similar fashion. While there is some evidence that blacks and Latinos are experiencing even more severe dysgenic pressures than whites, which could lead to further divergence between whites and other groups in future generations. What he means by dysgenic, what they mean by dysgenic pressure is that the people who have less ability genetically are reproducing at higher rates, and therefore the entire population is declining in their innate ability. So again, we first look at the cross-section. Indeed, as we know, in almost all societies, Bob was telling me that last night that there's actually an exception here among a particular subgroup, but higher education leads to lower fertility. That's known across, that's what's called the demographic revolution, that's a demographic transition, that's known across the world. That's not news. But we looked at, again, with the genotype, the effect is much smaller. It's still negative, negative association between educational genotype and number of children ever born, but it's much smaller. And it varies for the other for the other outcomes. Now, again, the questions about the trend, and again, if you look just at the phenotype, if you looked at just the raw education of somebody over the course of a woman over the course of the 20th century and the number of kids she ever has, you see that at the beginning of the 20th century, there actually was no effect. And by the end of the 20th century, there's a widening gap in fertility. And there's no effect, actually, except for BMI, it goes the other way. There was a fertility penalty for being overweight in the early 20th century, and that's gone away now. But again, if we look at the thing that we're concerned about, that we're transmitting, or increasing inequality or lowering the mean, in this case, across generations, over time, that doesn't seem to be happening. There's no significant effect. So again, we use the genetic data. We're not afraid to ask the same controversial, some would say, racist questions that Herrnstein and Murray asked, but we come up with very different answers when we use it, the state of the art. I'm going to skip over all this to talk about gene by environment interactions. So the reason that I think a lot of us social scientists who went into this area of genetics did so was because we witness that the fact that individuals respond very differently to the social environment. And sociologists are very fond of saying, well, if genes matter, then how they matter depends completely on the environment. And there on the left is a picture of Jimmy Carter visiting New York City of my childhood. This is a very worse neighborhood than I grew up in the South Bronx. And you can imagine if there's such a thing as a gene for aggression or a genotype for aggressive behavior, and you grow up in that environment, you're going to end up in jail, right? That on the right is the house of being refurbished of Mitt Romney, the 2012 presidential candidate, and almost not quite a billionaire, but very rich. If you have the same genes for aggression that he might have, and you grow up in an affluent suburb in the United States, you might end up as CEO. The same traits, same genetics plays out very differently depending on your environment. That was the promise of why we went into this to integrate this. So what do we find? So it's very hard to find what we'll call exogenous environment. In the social world, the equivalent of a medical study where there's a treatment group and a control group, and people don't choose which to be in, and therefore we can actually know the effect of the environment. That itself is very hard. And then we're adding to that, we want to understand how that varies by genotype. So here is the man I used to call the dumbest politician in the history of the United States. I have to change that after last November. He is a representative who thought it was a good election strategy to get publicity, pulling the draft numbers of his constituents' children, sons, out to send them to Vietnam. This was a literal lottery that was held in the late 1960s, in 1970 in the United States. And it was it's a horrible national experience to sit in front of the TV and watch this. But for social scientists, it's a great natural experiment. It's been used for a variety of outcomes. Jim Heckman has criticized it I won't get into all the nitty-gritty details of why it's why or why not it's the ideal natural experiment. But for our sake, it's one of the few that we can actually look at in our data. So again, we asked whether or not being drafted, randomly assigned to being drafted, affects your health in this case. We look at number of cigarettes smoked per day, and we know that the Army, I don't know how it is here, I'm sure you're healthier, but in Vietnam in the 1960s, the military was a very pro-tobacco environment. Cigarette smoking was rampant. In fact, cigarettes were made very cheap or free as a stimulant to the troops in combat. So many people took up the habit when they went to war. So that treatment of being drafted is a treatment of likely becoming more a smoker. But what we find is not created equally here. So if you're at high risk, as I mentioned, before in terms of the trends over time, if you're at the upper end of the distribution for genetic risk for becoming addicted to tobacco, then getting drafted has this huge effect on your lifetime smoking behavior that increases your smoking per day by a pack of cigarettes per day on average. If you're at the upper end of the distribution, 2 standard deviations above the mean or higher. Now that's a huge effect, a pack a day. That has incredible translation to lung cancer rates and so forth. But if you're not at genetic risk for becoming a lifetime smoker, being drafted, it's not going to affect you at the left end of the distribution. Well, the draft also had important effects on education. It made veterans eligible for what's called the GI Bill in the United States, which is basically a scholarship to go to college, but it also took people out of their educational careers, so we don't know what the net effect is. When we look across the education genotype, we find that at the negative, that the low ability draftees suffered educationally. They end up getting less schooling than their non-drafted, exactly genetically equal counterparts, but who are not drafted. So there's a gene by environment interaction. So what you can do with the genetics is you can see how a certain policy is going to have differential effects in your subject pool or in your population. And it acts, as I would say, as a prism to refract the white light of one treatment into a rainbow of different responses. If I have time, I'm going to go on to one more part, which is really the whole notion that of genes and environment as a sort of wall between them or as two distinct realms is being challenged now with these new genetic data and new genetic findings. So first, I'm going to show you evidence about genetic distributions within the family. So this is from a different study. It's called the Minnesota Twin Family Study. And you can see on the far left, in the blue bar represents the effect on an individual's educational attainment based on in standard deviation units, based on a 1 standard deviation change in their polygenic score that I've been talking about. So a 1 polygenic score, a 1 standard deviation increase in a sibling's polygenic score leads to about 2/3 increase in their standard deviation increase in their schooling. But what we find is that even when we control for the parents' education and their genotype, that the siblings' genotype influences myself. So my sibling, because again, we're only 50% related, my sibling's genotype is not the same as mine, but if the sibling has a high genotype, 1 standard deviation increase in my sibling's genotype, net of my own, increases my educational attainment by 4/10 of a standard deviation. So, the genes of the people around us are affecting us as well. In a sense, they're part of our environment. When we dug into this a little more, if you look at the third set of bars, you see that when we look at sisters only, we see a big effect. Sisters influence each other more, that they're half a standard deviation increase in my sister's polygenic score increases my education. 1 standard deviation increase in my sister's polygenic score increases my education by half a standard deviation. Then we look at brothers, we don't find any effect of the brother. So there is a tool using genetics to interrogate gender relations within the family. And I think this informs or is informed by work of Carol Gilligan and others that suggest there's very different learning styles and different interactional styles, cooperativeness versus competitiveness by gender, and we can see this in the family using the tool of this independent random draw in the family of the genotype of myself and my sibling. And I'll show you another example of that. One of the long-standing concerns in sociological research and in education research is what is the influence of peers on my behavior. But that's notoriously hard to study. You have the fact that if we measure who I know, my peers, my friendship network, we have the phenomenon we call birds of a feather, or the fact that, or homophily is the formal term, that who I'm friends with is not a random assignment. I select people like me, they select me, and therefore if peers are like each other, how do you know that they weren't already like each other and then just became friends, or if they're actually influencing each other to become more like each other? Then you have what economist Charles Mansky called the reflection problem, which is, how do I know that because Yossi and I are friends, whether he's influencing my behavior and choices or I'm influencing his, if there's a correlation between us? It's very hard to separate out who's influencing who. And finally, you have what's called contextual effects. How do we know if it's the impact of something that affects all sociologists, from externally affects all sociologists and not economists, or vice versa, or people on the left-hand side of the room or the right-hand side of the room? We don't know that there's not some third force out there that's causing peers to behave similarly, and rather than them actually influencing each other. So genetics can actually help us here too, because everybody brings their polygenic score to the social group independently, if you do it right. And this is what we do. We look in the National Longitudinal Study of Adolescent to Adult Health, and we look within each school. So yes, you might think that people genetically sort, as we were hearing much about earlier, into different neighborhoods and different regions of a country, so they might not be equal genetically when they come to school, but within a given school, whether or not there are more people who are prone to smoke, let's say, genetically prone to smoke or to get high grades, in 7th grade versus 8th grade of that school is random. That's what we call that the treatment, and this allows us to understand peer effects. So in this case, we look at smoking, and we find that there is a very big peer smoking effect. In fact, the effect of the genetic distribution of your peers' likelihood to smoke or their actual smoking behavior has a bigger influence on you than your own genetics in this case. Again, kind of confusing the idea of nature and nurture. As I said, this is an interesting social tool. You can use this as a microscope to look at social relationships, independent of whether or not you're interested in genetics at all. So we look at race and peers here, and we find that the biggest effects are for whites with their white peers. When the white peers are highly likely to smoke, the white respondents have a much higher likelihood of smoking. If you look at whites with all peers, the effect is smaller. So in other words, whites are paying more attention to other whites' behavior. That's what in their grade. So that's something we learn through applying this genetic tool. And the peer effects for other races are smaller, as you see at the bottom. So to wrap up, why should social scientists care about genetics? Number one, as I started with, the genetic main effects have typically been very small. We have to add up these small little effects to get polygenicity, to get the polygenic score that has big effects. And that allows us to go back to the studies like of parental income or of an educational intervention like lowering class size or what have you, and get more precisely estimated effects and get less biased effects by factoring out any differences in genetic predisposition. We can also think of the social genome as an instrument or as part of the environment and try to understand, use it as a tool to understand how we influence each other within the family, within the schools, and so forth. And then we can do gene by environment studies. There's a hypothesis called stress diathesis hypothesis or differential stress hypothesis, which suggests that some kids are like dandelion flowers. Basically, they look the same. They grow anywhere, sand, good soil. A dandelion looks like a dandelion. It can grow in many parts of the world. And other kids are sensitive. They're orchids. They look beautiful if they give them the perfect humidity and temperature and sunlight combination. But if they don't get that, they wither and die And so the intuition, again, is that genotype explains why some kids are susceptible to stress or poverty and some kids are resilient, and that we could better understand that and target that, if you want, on the part of policy. And I should say, it's not that one kid is a orchid for all outcomes and one kid is a dandelion for all outcomes. People have different risks. They may be an orchid in terms of addiction and depression and mental health, but they might be a dandelion in terms of body mass index, diabetes risks, and so forth. So this is a controversial area. I also mentioned that we could take a few cells from a fertility clinic, from an embryo that's going to be implanted for a couple that's trying to have kids through in vitro fertilization, and if they have 10 embryos, we can say which one would be the tallest, likely, which one will do the best on tests in school, and so forth. And I think we need to think that sounds like science fiction. In fact, there was a movie in the mid-1990s in the United States, again in this strange time, called Gattaca, that presciently predicted exactly this kind of self-generated genetic selection. And that's an important policy discussion that I think we have not been having, whether we want to allow that, whether we want to facilitate that and democratize that, because right now I can predict that in 5 years, rich parents will be doing that. And the unlevel playing field will not just be in school, it won't just be in the cradle or the womb, it'll be in the IVF laboratory if we don't do anything about it. Maybe we'll talk about that in 5 or 10 years at a conference here, hopefully not. There are certain societies like China right now that is explicitly trying to do this, trying to sequence high IQ so that there's no worries, as there is in the Judeo-Christian world, about playing God in Confucian or communist China. As we know, they were very successful in a one-child policy. I think we should pay attention to what's going on there in terms of this kind of policy as well. So I think it's really important to have social scientists and policy experts at this table of this genomics revolution and part of the discussion, because if we're not, it's still going to go ahead without us and have enormous implications for educational inequality going forward. So thank you very much.
My name is Dr. Michael Schneck. I'm a semi-retired psychiatrist who made Aliyah to Israel in 2005. I've read a lot about genomics. There's lots of things that change every couple of years in genomics. I've never read about a polygene score, certainly not in behavioral field, in psychiatry or otherwise. I also read a lot of oncology genomics. What is it, is there, I mean, everybody, what I read is that complex behavioral traits or complex medical problems are all too hard to discern, much less to then take an environmental gene interaction. What is, most of what you said is based upon polygene score, right? Is there much data to support that?
I mean, I'm not sure.
That exists?
That what exists?
That such a thing as a polygene score really exists.
I mean, I don't even know how to answer that. Yes, it exists. That's what I've been talking about for the last hour.
Right,
and,
I've listened, but...
I mean, I can refer you to many papers that have looked at psychopathology. All of these come out of consortia because no single study is big enough to run the models to get a good polygenic score on its own. That's why we combine data from 23andMe, the UK Biobank, Swedish Twin Registry, a whole number of studies to get that 1.18, I think it was, million sample. There's a group called PsychGen, that it's a consortium that does psychiatric genetic outcomes, so psychopathology, and they have a number of scores that predict psychopathological outcomes: schizophrenia, major depression, the big 6 major illnesses. It turns out that actually the score that predicts any of the 6, whether you have any psychopathology, is the best predictor for any of the 6, which is interesting. That means suggests there's a common basis for mental disorders.
Please go ahead. All the way in the back, though. In the back over there. Yeah.
Thank you very much for the talk. Josiah Rothenburg. Two questions, I think. One is, and I apologize, it's been a long time since my stats and econometrics courses, but I think early on in the talk you said the R-squared was 13% for the polygenic score, and so that's explaining about 13% in the variation, and that was the largest predictor, at least amongst the variables that you.
It wasn't the largest. 13%. I should say two things. 13% incremental R-squared. R-squared above and beyond control variables. Second, it wasn't the largest. Parental education itself is the largest single predictor, but it's I think it was 2/3 of that size.
So with that, and I guess it depends on which side of the argument you're on. To me, to the extent that genes are, or non-environmental factors are really important. That seems to me, ultimately encouraging, meaning that most of environment is most, right? And despite and I don't mean to
no, I get
your career, right? But
I get what you're saying. You're saying that 87% is environment, 13% is genes in that case, which is good news. But that's not. I'm sorry to rain on your parade, however, on your optimism. But that's 13% is an underperforming. There's a lot of measurement error. We really need at least a 3 million person sample to achieve the full predictive power of these common variants. It's estimated that's about 25%, actually, if we get to the full predictive power, and that full genetic effects are on the order of 40%. Now, I want to also challenge the sort of assumption that environmental effects are good and genetic effects are bad, because imagine the world, if we talk about justice as fairness again, imagine the world where common environment of your household environment matters a lot. That's not a fair world. That's that irregardless of my ability, the family into which I'm born in, a rich family or a poor family or a rural family or an urban family or whatever, has the lion's share of determination of how I'm going to end up in society. That's not a world we want. I mean, some more radical thinkers argue that we want a world where it's 100% genetic, because any environmental influence is pernicious. It's not fair, and that based on ability per se, is a fair meritocratic world. Now, I don't go there, but I do want to challenge the assumption that lowering the genetic effect and increasing the environmental effect is the policy goal, because that's not necessarily. And I think we come back to the notion that you want equality redistributed, regardless of what the effects are. And I meant to say this earlier about Avi's comment about policy. Avi is wearing glasses. We know that myopia is highly genetic in modern society. And I had LASIK eye surgery myself. You're wearing glasses. We know, and the economist Arthur Goldberger made this point back in the 1970s, that just because something's genetic doesn't mean we can't fix it. We have a perfect fix for myopia, very cheap. What it means is that if you fix it for Avi, it doesn't automatically fix it for the kids. His kids are probably going to have to wear glasses too. We're going to have to keep fixing it across generations. If it was purely environmental when we fixed it, then maybe it would be transmitted to his kids and we wouldn't have to give his kids glasses. But that's all it implies for policy, that there's no dynastic policy effects across generations. It does not imply that we can't do anything, and we should not do anything, I would argue.
מיקרופון, אולי תדבר בקול רם, speak up and we'll save the mic. Oh, translation can't hear, sorry. Pass it back, yeah.
First, it was a really fascinating talk, and just a technical, but also a question, but also has some larger meanings. From what you say, it seems like the polygenic score doesn't take into account the interactions between the locations. Now, if you take that, maybe even you will find out that actually genetics accounts for 40%.
That's a good point. There's lots of work going on, and what you're talking about is that this is a purely additive model, and there might be interactions, what's called epistasis in genetics, that gene-gene interactions, like having the C there and the T there is the particular combination that matters, not whether you have a C if you have A, or you get it, what I'm saying. But the estimate is that most of the effects are additive. There's evolutionary reasons for that, that it's very hard to design for evolution to happen if there's a lot of epistasis, or a lot of because then you're basically trapped. You have to have this unique occurrence of a mutation that's beneficial in two different places at the same time in the same organism, the same generation, for it to be a better fitness. But the estimate is maybe 10 to 15% of what's called missing heritability might be due to gene interaction, and also from gene-environment interactions, because we estimate these things across different environments, but how the environment is matters what genes matter as well. That's one of the important findings.
When you compare the parent, especially the mother education, and the gene influence, how do you know you're not measuring twice the same thing, that actually it overlaps? That's a very good point, that if people heard the question, that in a sense, I was showing those bars about mom's education because it's a very typical thing that social scientists will put it. In fact, it's the most powerful predictor in social science equations, but part of that mom's education is itself genetic, and in fact, there's new papers out that show there's what's called genetic nurturance. So the genes that your parents have, I already showed the sibling effects, your parents' effects are even stronger, that even the ones they do not transmit to you form part of how they behave and therefore affect you. So it's part genetic nurturance. So there's a famous claim by the psychologist Eric Turkheimer from the University of Virginia that says there's a gene-environment interaction that, so low socioeconomic status families, there's not a lot of genetic effects. High socioeconomic status families, there's big genetic effects on IQ and education, because the theory is that in low socioeconomic status families, there's not enough resources, and the environment is so poor that people don't even get to differentiate themselves based on their genetic propensities. However, when you have a lot of resources, then you see differences emerge. That was based on twin studies and a number of dubious assumptions, I think. We reanalyzed that question using the polygenic score and found that there was no kind of SES, mom's SES, child's genetic score interaction. What the real interaction was between the mom's polygenic score and the child's polygenic score, and they mutually reinforce each other.
Thank you very much. So the question is this. You talked about the role of polygenic score in determining the outcomes and helping us predict the outcomes. And you talked about the entire population, but we're talking about inequality in education, and I'm wondering whether this relationship holds also in the left and the right tail of the distribution of education. So if I would go to Princeton and I would try to measure this, what is the evaluative prediction? What is the value of the prediction of the polygenic score on the success in Princeton students in comparison to other factors such as income and education? And more importantly, if I would go to America Super Zips and I would see what is the value of prediction of the polygenic score in determining whether you can get into Princeton or one of the Ivy Leagues, will this still be a very strong component in our prediction? And I'm asking that because in the winner-takes-all society, looking at the top end of the distribution matters a lot, and I think that this is part of the concern that we're talking about. You can also say similar concerns about the left side of distribution.
I mean, that's a very good question. What I'm talking about are effects around the mean. Of course, it's estimated through linear models. I don't think we have the statistical power right now to do specific quantile of regressions. However,
I'm sure that's coming. I will say, however, that when we've looked at these interactions, crudely cutting the data, we don't see the difference in effect, at the top half or the bottom half of the distribution. In general, it's very hard to get large samples of elites, for example. However, in the genetic world, because a lot of people who participate in these studies are very highly educated, like 23andMe data are people who pay money, send their saliva in to get their own results. They're very highly educated. UK Biobank is people who agree to participate. They're more educated than the British average. So actually, we have truncation bias. We have an underestimate of the effect because we're estimating on a truncated part of the distribution, where people who have college versus a post-college degree, or we're not getting the left-hand side of the distribution. So there's a lot of estimation issues that I think people are making progress on.
Ken, I'm relating to your remark about the lower birth rates at the higher educational classes, and I'm combining it to the low birth rates all over Europe. Actually, Europe is committing suicide. And I'm asking, my question is, are you concerned?
The policy, I'm for open borders. So if you're for open borders, then you have no birth rate problem. Only if you're a nationalist and you care about the French dying out or the German Volk dying out, then you care about birth rates. Because if you don't, if you care about just economic activity, just allow more immigrants. So there's plenty of places that have overpopulation. We can just equal it all out.
But how is your genetic theory combines with that?
Whether or not we're having differential birth rates by ability. I mean, it's not just me. There's other estimates that do show from Iceland, from other countries, that the average polygenic score is slightly drifting downward over decades. Very small effect, however, compared to what's called the Flynn effect in psychology, that IQ has been rising over decades, at least of the last 100 years, probably due to there's no lead in gasoline, there's no lead in paint, there's abortion, so unwanted children are less I mean, there's a whole number of effects. We live in a more complicated society, so maybe we're learning to process information quicker. So the good news is that cognitive ability is actually increasing by decade.
Okay, I'm told we have time for 2 more questions. Please go ahead. Person with the,
okay, so my name is Herbert Rosenfeld, and when you were talking about the bell curve, I was thinking about somebody else you could compare your work to. I'm just very curious, how does your work compare to the work of Arthur Jensen? I mean, he basically said there was a general cognitive ability that's essentially inherited trait, and that was the first way that his research was understood. I wonder what you think about that. And the other thing, which is the elephant in the room, no one's talked about it, what about race and intelligence?
Okay, the first question is, I mean, Jensen and Herrnstein and Murray are twins. I mean, Jensen's the progenitor of Herrnstein and Murray. So they're on the same, I mean, I test their hypotheses. I don't find strong evidence for it. So Jensen's included in that group. The second question about race, you notice, I avoided race this whole discussion. There's 2 reasons for that. The main reason is technical. So if you want to know about the single biggest genetic difference in the world, it's between people of African descent and people of non-African descent. The folks that migrated out of Africa, approximately 100,000 years ago, the exact date is debatable, were
dwindled at one point to somewhere between 1,000 and 2,000 people. So much of, when you have a population bottleneck like that, much of the genetic variation is eliminated because whoever just survived, they carry their genes forward, and all the gene variants that didn't come forward don't come through the population. So today, I'm half Ashkenazi and half European mutt, as we say. And I'm as related to a Han Chinese, as genetically speaking, as a Fulani and a Hazda herder, tribe that are about 400 kilometers away from each other in East Africa. Sub-Saharan Africa is the well of genetic diversity. So why I'm going into all of this is because what's called the haplotype structure, or the genetic structure, is very different for people of African descent than of non-African descent. So when we have these large samples, where are they coming from? They're coming from Sweden, the UK, Australia, the United States, other Western European countries. Yes, they have some minority people of African descent in those populations, but the vast majority of European descent. Their genetic structure, so when I talked about the T or the C at a particular location, it's not that change in particular is causing a protein to be different or causing a protein to be expressed more or less in your brain. It's just a marker for that region of the genome, for the whatever 1,000 base pairs above and below it. And Europeans have a common block there. So when I measure C or T there, I'm measuring the same thing when I measure European populations. African populations have much more variance because of that greater genetic diversity within that block. So when I measure C or T, I don't know what I'm measuring in that block. And in fact, we try polygenic score prediction on African people, either African Americans, for example, or Latinos, who are a mix of Asian, European, and African genomes, and they don't predict. And I don't just mean IQ or something, or education, I mean height. So the height polygenic score, which is an incredible predictor, you could predict your baby's height very well. Of course, you could probably predict it from the parent's height too. But it predicts African Americans to be 6 inches shorter than they actually are on average. So it just doesn't, the genetic structure doesn't work to translate. But that implies that there is actually implicit, kind of an implicit racist dynamic happening because of who has the samples. We have large samples of people of European descent. We have polygenic scores for people of European descent. We do not have them for those people of African descent. So if there actually is a policy or a medical benefit that comes out of this, it's disproportionate for whites. There's NIH in America, National Institutes of Health, is aware of this, and there's starting to be calls for major genotyping studies of African Americans and other minority groups so that they can have their quote-unquote own polygenic scores. So it's just not possible to do it across, and even if I tried to apply, it wouldn't, and you wanted to ask a racist question, you couldn't, because it's like comparing apples and oranges genetically. It's not the right measure.